Okay, so welcome to this next video in which we're discussing the antithrombotic mechanisms of the endothelium. So far we've seen a variety of mechanisms for preventing the formation of a thrombus within a blood vessel. We've seen how antithrombin 3 will bind to heparan sulfate proteoglycan, which is on the surface of the endothelial cells, and that will activate the antithrombin 3, which will then inhibit thrombin 9A, 10A, 11A, and 12A. And if these are inhibited, then you're not going to get coagulation either through the intrinsic coagulation pathway or the extrinsic coagulation pathway. So you're not going to get the production of fibrin strands. And since fibrin strands are essential for the production of a viable thrombus that's actually sewn together properly, then if you don't get coagulation, you don't get you don't get thrombosis, basically. Okay. In addition, we've seen thrombomodulin, which binds to thrombin. Okay, so thrombin will bind to thrombomodulin, and then its function will be completely changed. It will now activate coagulation factor 14, also known as protein C, which, when that becomes activated, is going to combine with protein S, which is a cofactor for it, and that enzyme will then degrade factor 5A and 8A and inactivate them. Okay, so those won't be active, and those are essential for both the, 5A is essential for both the intrinsic and extrinsic coagulation pathway, and 8A is essential for the intrinsic coagulation pathway. So by inhibiting these, we're going to stop coagulation again. Okay, so both of these pathways are aimed at stopping coagulation, stopping the production of fibrin strands. Then we've got mechanisms to stop platelet aggregation. So we've got ADPases, which break down ADP to AMP and inorganic phosphate, and if we destroy the ADP, then we stop the chain reaction of platelets activating other platelets. So we stop the production of a huge amount of thromboxane A2. And since thromboxane A2 is necessary for the vasoconstriction and also for the activation of the GP2B/2A protein, if we don't have thromboxane A2, we're not going to get vasoconstriction of the blood vessel or platelets becoming sticky, so we're not going to get platelet aggregation. Then finally, the endothelial cells we've also seen produce nitric oxide and prostacyclin, both of which cause vasodilatation, counteracting the effects of thromboxane A2, which is released by a thrombus. And in addition, prostacyclin also prevents activation of platelets. If a prostacyclin molecule is bound to the prostacyclin receptor on a platelet, then this platelet will not undergo activation. So even if it finds collagen or tissue factor or ADP, it won't undergo the activation response where it starts chucking out ADP and thromboxane A2 itself. So this will stop the production of thromboxane A2. And therefore will stop platelet aggregation. In addition, nitric oxide stops the effect of thromboxane A2 on actually activating the glycoprotein 2B/2A protein, and therefore again stops platelet aggregation. Okay, so now the final little thing that endothelial cells do. Okay, so they also produce plasminogen activators, and they produce two plasminogen activators, which I'll tell you about. Okay, so here's our endothelial cell, and it's going to produce two enzymes which are going to be put on the surface of this endothelial cell. Okay, so here are these two enzymes, and I'll put the nucleus there to make it a little bit more obvious that this is actually a cell. Okay, so one of these enzymes is known as tissue plasminogen activator. Okay, so, or TPA for short. So this stands for tissue plasminogen activator. And this tells you exactly what it's going to do. It's going to activate plasminogen. And I'll tell you what plasminogen is in a moment. So tissue plasminogen activator. Okay, whereas this other one is going to do the same thing, so we might as well discuss its name first. So this is got the name urokinase, and it's abbreviated to UK. Urokinase. Okay, it's also known as urokinase plasminogen activator. Its full name is urokinase plasminogen activator, so you may well see it abbreviated to UPA for short. Okay, so what do these two enzymes that we've produced and put on the surface of our endothelial cell actually do? Okay, so here is our tissue plasminogen activator in pink, and then we'll have our urokinase plasminogen activator in green here. Well, basically both of these enzymes are going to activate plasminogen. So if we've got plasminogen here, plasminogen is a component of the blood. Okay, so it's produced in the liver again, and it's the inactive enzyme, okay? So plasminogen is circulating within the blood, but it will be activated by tissue plasminogen activator, or indeed by urokinase plasminogen activator, and it will be turned into the enzyme plasmin. Okay, and now what does the enzyme plasmin do? Well, plasmin degrades fibrin, so it's going to catalyze the breakdown of fibrin strands. So these polymers of fibrin which hold together a thrombus, or indeed a hemostatic plug, and it's going to break them down. Basically, it's an enzyme that's going to chop up the fibrin strands. So here is a fibrin strand, let's say, in pink. What it's going to do is it's going to break it down into loads of little pieces. Okay, and these are known as fibrin breakdown products. Okay, so this is a fibrin breakdown product. So what does plasminogen overall do? What's the big picture? Well, if you break down fibrin, then you're going to break down the support meshwork for a thrombus. Okay, so once you've broken down all the fibrin meshwork for the thrombus, what's going to happen is the thrombus is just going to break apart, because I've told you that the adhesions between the bonds between the platelets are very, very weak, basically. So these interactions between GP2B/2A and fibrinogen, and then another GP2B/2A. So let me draw this. So here's a platelet, and then we have our GP2B/2A, and then a fibrinogen molecule in between, bound to another GP2B/2A on another platelet over here. These interactions are very weak, basically. Okay, so this isn't strong enough to hold together a thrombus on its own. So when you break down the fibrin, that's going to break down a thrombus. So if you do actually create fibrin strands, if coagulation does get active, plasmin is going to always be there active to break down the fibrin strands into fibrin breakdown products, and this is going to stop the formation of thrombi. Okay, so that's the final mechanism that endothelial cells have for preventing thrombosis. They have these tissue plasminogen activator and urokinase plasminogen activator enzymes on their surface, and these are continuously converting plasminogen into plasmin, which will break down any fibrin that is any fibrin strands that actually are being formed within the blood, and stop them from actually assembling into a thrombus.